Hello and welcome to Angiogenesis, where I simplify biochemistry. My name is Samruddhi Deshpande and I am a medical student at Dr. V.M. Government Medical College, Solapur. I love biochemistry and I hope through my videos you will like biochemistry just as much as I do. So let's begin with the first chapter, Heme and Hemoglobin. I will be covering this chapter in a series of 6 videos. The first will contain heme and its synthesis, the second disorders related to heme synthesis, the third will be heme catabolism, which is basically the production of bilirubin, the fourth part will be disorders related to bilirubin, which is essentially jaundice, the fifth part will be the transport of oxygen in blood, and the sixth and the last part will be hemoglobinopathies or the disorders of hemoglobin. So stay tuned. So to begin with the first parts, let's just first study what RBCs are. We all know RBCs are biconcave cells, 7.8 microns in diameter, all of that basic knowledge. So these are cells which transport oxygen in our body. These are cells that transport oxygen. Their main function is the transport of oxygen. Now, to make space for more oxygen to be transported, the nucleus and organelles had to be removed. That's why they don't contain any nucleus or organelles. Instead, they contain this protein, very important, called hemoglobin, which we will study about later, which carries oxygen. Now, you might wonder, where does the ATP and NADPH, which is required importantly, comes from? So the NADPH is obtained from the HMP shunt pathway, which we'll study in carbohydrate metabolism, and glycolysis, which we'll also study in carbohydrate metabolism. So both of which do not require any organelles and both of which happen in our cytoplasm. They are the most important sources of NADPH and ATP. And so naturally RBC will only depend on glucose for its energy sources. So no other, like fatty acid or ketone bodies can be utilized by RBCs, because mitochondria is absent, which mainly mitochondria is responsible for converting these into energy. So RBC fully relies on glucose. Okay, so heme, let's study heme. What is heme? Heme is a coordination complex. Okay, you might have learned in chemistry in 12th standard coordination compounds. So there was a metal, and then there was the D shell, and there were unpaired electrons. So similar thing is happening here. You have unpaired electrons in the D shell of iron, right? 5 there are. So there are 6, So there are 6 valencies of Fe to be satisfied. And how do we satisfy this? With some compounds which we'll study later, which are called porphyrins. Okay, so Fe is coordination forming with 6. Fe has a coordination valency of 6. So Fe is forming coordination bonds with 6 compounds. And that's why heme is a coordination complex. Now it has porphyrin. What is porphyrin? It's a tetradentate ligand. Okay, what is meant by tetradentate ligand? That the complex that Fe was forming, 4 out of 6 are covered by a single porphyrin molecule. Okay, so the porphyrin satisfy 4 of its valencies and 2 are satisfied by others, which we will study later. So basically heme is a coordination complex of iron which has porphyrin. The other 2 I'll tell you, it's H2O, it means water, and there is histidine. Okay, so we'll study about that later. But these are the 6 entities that satisfy its valency. Okay, now heme. So we got heme, that is a coordination complex. Now we got heme, and now heme will form compounds with, say, proteins, polypeptides or stuff like that, to form our hemoproteins. This is a family which is called hemoproteins. Now this family contains hemoglobin, which is the most, physiologically obviously the most important form. Others include myoglobin, cytochrome, catalase, peroxidase, and nitric oxide synthase. Now I'll explain. So see, I will explain these by their functions. Okay, so functions of heme are, so the first is transport of gases. How? Hemoglobin, obviously hemoglobin will transport gases, CO2 plus O2. Very simple. ET chains is electron transport chains. Electron transport. Where does this occur? Electron transport occurs in where ATP has to be produced in the mitochondria, on the membrane of the mitochondria. So for this, what we use? Cytochrome. Do you remember cytochrome C, which was the mobile protein which moves electron? So for electron chains, heme is required. Then next is chemical catalysis, which will break down compounds, which we will study about in other chapter. But just for an idea, chemical catalysis and antioxidant defense. Peroxidase and N-synthase both are involved in our antioxidant defense. How? We'll study in antioxidant, but I'll give you an idea. Peroxidase will convert H2O. It's a whole cycle, but we'll study about it later. Anyway, now these are the functions of heme, okay, and they correspond to the family of hemoproteins that we just studied. Now hemoglobin, because the most relevant part here is hemoglobin. So hemoglobin is a conjugate protein. We'll see why it is called conjugate. Why? See, it has two parts. It has a heme and it has a protein. So there are two parts which come together, which conjugate. So that's why it's called a conjugate protein. Now, to give you an idea of the structure of hemoglobin, I'll just try to give you an idea. So there is heme and there is prosthetic group, okay, and it's tetrameric. So let me explain. Tetrameric has 4 subunits. Am I right? Okay. So now it has 4 subunits, which means that it will have, let's say, these are its 4 subunits. Okay. Now each subunit has a heme and a protein. It's like distributing to each child, There are 4 children and you have to give each one heme and one protein. So I give you one heme and a protein. Heme protein and heme protein. Okay. So like I'm giving each two subunits. Like there is a heme and there is a protein. So to give you a graphical idea, let's say these are heme and each one has a chain, like a polypeptide chain. Okay, so this forms our hemoglobin. This is just a graphical idea. So now this protein chain, it can be different, obviously. Now there are types of hemoglobin, like there is fetal hemoglobin, there is HB1, there is HB2. But the most common hemoglobin has 2 alpha and 2 beta chains. Okay, let's say these are 2 alpha and 2 beta chains. So this forms our hemoglobin. That is the basic idea what hemoglobin is. We'll study about it in the next videos. Okay, so now that we studied hemoglobin, let's move on. Let's just study in more detail heme. Now structure of heme. It's very important and also a little complex. So now heme, okay, it's a porphyrin derivative. Okay, just stay with me on this. Heme is a porphyrin derivative. Okay. Now, porphyrin derivative means heme is formed from porphyrins. Now, what is porphyrin? Porphyrin are cyclic compounds. मतलब porphyrin है, they are cyclic. मतलब porphyrin ऐसे समझो कि they are cyclic structures, ठीक है? And they something उसमें करके we are forming heme. Now, porphyrin किससे बनता है? They are formed by pyrrole rings. Okay, I know, very complex. So tentatively you just remember for now कि there is basic structure called pyrrole rings, then they form porphyrin and then they form heme. So pyrrole rings क्या होते हैं कि just think of it like polymerization, right? Pyrrole ring, they are monomers and then they say polymerize to form porphyrin, and for porphyrin से जब हम Fe जोड़ देते हैं then we get heme. That is it's as simple as that, right? So 4 pyrrole ring मिलके एक porphyrin बनाएगा. Okay, the exact structure of porphyrin is this. This is the exact structure. These circles are side chains. I'll discuss them. So see, this is one pyrrole ring. This is a pyrrole ring. This is a pyrrole ring. Okay, now there is conjugation inside the ring, but this is a pyrrole ring, and this ring is connected. You can see this is connected by these are bridges, no? These are sort of bridges. So these are methinyl bridges. Methinyl bridges. Okay. So to give you a very easy idea, let's just say these are 4 pyrrole rings. Okay. These are not circles. This is the exact structure, but I'm giving you a simplified structure. This is pyrrole rings, say, and these are connected by bridges which are called methinyl bridges. Now methinyl looks like this. Methinyl is this. Okay, this is the methinyl group. It's attached to a double bond and then a single bond. So this is a methinyl group. Now these are named alpha, beta, gamma and delta, but you don't need to remember that. It's just not that important. Okay, now this is the first part. Now what happens is, on each pyrrole ring you can see there are these side chains. Each pyrrole ring has 2 side chains. Side chains, by side chains I mean small carbon containing linear chain. So these can be methyl, right? These can be propionyl. Propionyl looks like this. See, it's like propionic acid. So it's CH2 CH2 COOH. Okay, this is what it looks like. So then you can have vinyl group, which is CH double bond CH2. The double bond is between the first and second carbon. And then also you can have acetyl group, which is just like acetic acid, which is CH2 COOH. Okay, so these are the groups that you can have: methyl group, propionyl group, vinyl group, acetyl group. So these are named M. You can also M, P, V and A. These are the groups that you can be present here. So based on these there will be types, of course. Now these types can be, say, you can have M methyl or propionyl, or you can have propionyl or vinyl, or you can have vinyl or acetyl. There can be combinations, right? But there are some fixed combinations that are found in our body. Okay, और इन कॉम्बिनेशंस पे बेस्ड देर आर टाइप्स और सीरीज. सो टाइप्स में हमारे पास कौन से टाइप्स है? टाइप 1, टाइप 2, टाइप 3, सो ऑन. एंड सीरीज भी होती है जिसमें 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 ऐसी सीरीज होती है. ओके, सो the most biologically important series or type that you have to remember is type 3 or 9th series. This is the most important form, which will finally produce heme, which will see in heme synthesis. So type 3 or 9th series, which is the asymmetrical series. Okay, you don't have to know why, but just asymmetrical it is. The arrangement of side chain is asymmetric. It's MP, MP, PM, MP, PM, something like that. And the type 1 is the symmetric series. Okay, that was the basic structure of heme. Now let's discuss synthesis of heme. Now heme synthesis is obviously very essential because it produces hemoglobin, which is responsible for the transport of oxygen. So you can guess how important it must be, right? So now it is a compartmentalized pathway. Now what I mean by a compartmentalized pathway is that it occurs partly in cytoplasm and partly in mitochondria. That means कि some steps occur in cytoplasm and some steps occur in mitochondria. Now there are a few other pathways in the body which act like this. Now if you want to remember, it's the hug pathway. You can remember like that. H is heme, U is urea cycle, and the G is gluconeogenesis, and the P in the pathway is for pyrimidine synthesis. So these are the 4 pathways that occur partly in cytoplasm and partly in mitochondria. Okay, we have to study all these cycles, but let's see heme synthesis first. Now heme synthesis, where does this occur? Obviously, like we discussed, some in partly in cytoplasm and partly in mitochondria. Now in what cells it occurs? All tissues can produce heme. All tissues can produce heme because RBC, because there is not much machinery required. Now you might ask, why RBCs do RBCs produce? Now I just told you that some of it occurs in mitochondria. So it's impossible for RBC to produce heme, right? So this occurs in normoblast cells. if you know, if not heard of it, I'll tell you. See, there is a process called erythropoiesis. There is a process called erythropoiesis, which is the production of RBC, right? Now, the RBC are not formed directly from, you know, stem cells. There are processes. There is a pro-normoblast, there is a early normoblast, there is a normoblast, there is a late normoblast, there is a reticulocyte, and then there is RBC. So the normoblast stage, which has organelles, okay, organelles are started to, you know, regress in the late and reticulocyte stages. So before that, the cells will still have organelles. So normoblast cells can produce, right? So they produce heme, and this heme is converted into hemoglobin. This is a very important point, because RBC, it's impossible for RBC to produce hemoglobin, heme, right? Now, the steps, let's see the cycle. This is the tentative cycle, which is very simple, looks a little dangerous, but it's very simple. Okay, so let's start. So the initial what you can say is step is the reaction of succinyl CoA and glycine to form delta amino levinonic acid, which is also called as ALA. This is the rate limiting step. Obviously, this is what is rate limiting step. It's the slowest step, and it determines how the rest of the cycle goes. So this is the rate limiting step of the reaction, and obviously this is this level at which all the the cycle will be controlled, right? Because rate limiting step is always controlled allosterically, say, or genetically, it's the rate limiting step that is controlled. Now, aminolevinonic acid is formed from succinyl CoA and glycine, right? Now, this aminolevinonic acid, which we call ALA, what will happen? It will be dehydrated, which is removal of water. It will be dehydrated by the help of, obviously, ALA dehydratase. Now, to learn the enzymes, it's very simple, because they are what they do, right? So dehydratase removes water, deaminase removes amine group, cyclase, and then so it's not that hard to learn the enzymes, right? So water is removed, right? And the water removed forms porphobilinogen. Now please pay attention, because this is a monopyrrole. Now what I mean by a monopyrrole is that, see, pyrrole I already told you, right? Like here I told you what's pyrrole. A pyrrole is this structure is called pyrrole, right? So what is porphyrin? It's a tetrapyrrole, right? There is 4 types. In heme there are 4. There are 1, 2, 3, 4. There are 4 pyrrole rings, right? So it's a tetrapyrrole. So now we are just at a monopyrrole. Now we want to form a tetrapyrrole, right? Now obviously, since porphyrinogen is a monopyrrole, to form a tetrapyrrole we need 4 porphobilinogen. That's exactly what we do. We form 4 porphobilinogen. We remove an NH3 group from them and form a linear compound. This is a linear compound, remember. This is called hydroxymethylbilane, or HMB. This is a linear compound, very important. And then this will spontaneously cyclize to form uroporphyrinogen 1. Now the 1 here is the type of the series which I just discussed. This is the type 1, which is the symmetric, right? It's the symmetric form series. This is the first type. Now obviously this is a tetrapyrrole, right? This is a tetrapyrrole, and a tetrapyrrole is called as a porphyrin. Very, very important. So this is the first porphyrin that we got, right? This is the first porphyrin of the whole cycle. Now this will be acted upon by UPG3 co-synthase to convert it into type 3. Okay, we want to convert it into type 3, which is symmetric to asymmetric. Now this is the most biologically important form, because after this all of the compounds formed are for series 3, and the enzymes which also act only on type 3 enzyme. Remember this. So uroporphyrinogen 3 will be decarboxylated removal of carbon dioxide to form coproporphyrinogen 3. Coproporphyrinogen 3 will be oxidized by CpG oxidase to form protoporphyrinogen 3. Protoporphyrinogen again oxidized to form protoporphyrin. Now I know it sounds very complicated, but there is no other option than to either learn it or understand it. To understand, I'll tell you very simple thing. So CpG oxidase. Now CpG is formed, it will oxidize. What it will do, it will convert two of the propionyl groups we discussed here, right? Here propion, suppose there are two propionyl groups, they're not exactly in this position, they're something like here or here maybe, right? So now these two propionyl groups will be converted into vinyl groups. So instead of P here, now we will have V. Okay, that's what the first oxidation will do, and the second oxidation will do something similar, which you don't have to learn, right? So protoporphyrinogen 3 will be formed, from which our protoporphyrin will be formed. Now protoporphyrin is a generalized compound. Now what we will do is we will add iron, which is our kind of our game changer here, right? So iron will add, and this iron will convert protoporphyrin into heme, which is our final what we wanted, right? So in a nutshell, if I want to tell you, see, succinyl CoA basically gave ALA, which is the rate limiting step. This is the step you have to remember, right? Is the most important step. Now this requires, okay, very important point, this requires PLP. What is PLP? Pyridoxal phosphate. Pyridoxal phosphate. It's a derivative of B6 vitamin B6. Very, very important. That is why in deficiency of vitamin B6 anemia is produced, because heme ही नहीं है तो obviously hemoglobin भी नहीं होगा and oxygen transport कम हो जाएगा and that will result in anemia. So very, very important, B6, PLP, and PLP is required in this process as a coenzyme. Anyway, the next will be formation of a monopyrrole. So you don't have to remember the whole cycle. Obviously, if you want to remember the names, you can, but I'm just telling you in a nutshell. So it will be converted into monopyrrole. Now here the important point is that the ALA dehydratase is a zinc containing compound and it's inhibited by lead. This will be important when we study disorders which is called as plumboporphyria. Plumbo is lead. So when it's also due to lead poisoning, we'll see about that. Okay. Now the next thing is it's a monopyrrole. We converted a monopyrrole into a tetrapyrrole. Now the tetrapyrrole, first it was linear, then it's cyclized. This is linear, then uska cyclization hua to UPG1, then UPG1 to 3. Now 3 is the most important, I already told you, UPG3, CPG3 and PPG3, UCP. And then finally you got protoporphyrin, we added iron and we got our beloved heme. This is in a nutshell the formation of heme. So there is a concept called hematin. Now we know that generally Fe is present at Av plus 2, right? In hemoglobin, you know, when in heme this Fe2 plus is converted into Fe plus 3, let's say by some oxidase, right? Instead of heme, hematin is formed. This hematin is very important, you might ask. Obviously it's of no use, right? For transport of oxygen it's of no use, but it is very important in regulating the cycle, and we'll see how in just 2 minutes. Okay, so let's just move on to the regulation of this cycle. Now 85% of heme is produced in erythroid cells or normoblast cells, which is then directed into the formation of hemoglobin, and the rest of the 15% is formed in liver. Why, you may ask? Obviously heme was not only used for hemoglobin, right? It was also used for peroxidase, this is also used for catalase, this is for also used for detoxification, other metabolic needs of heme, right? So this is only 50% of the entire heme production, but it's still very significant. It gives the rise to P450. This is a miraculous compound, very important. It is for the detoxification of toxins and drugs and etc. It's very important, and it is a derivative of heme, right? So this one of the most important functions of heme, right? Then The regulation is by three mechanisms: repression, allosteric inhibition, and compartmentalization. Very, very simple. As the name suggests, repression mechanism is, it's at the genetic level, right? It's at the gene level. What happens is the ALA synthase enzyme is inhibited genetically by the heme. Heme inhibits ALA synthase itself. मतलब final product जो है, it will inhibit the enzyme, the first enzyme. So this is also called as negative feedback, right? This is also called as negative feedback mechanism, right? This is a negative feedback mechanism because जो product आ रहा है वही अपने production वाले step को inhibit कर रहा है. So this is a negative feedback. The second one is allosteric inhibition by hematin. Now I just told you what hematin is, right? In heme, when Fe2 plus is converted into Fe3 plus, hematin is formed, right? This hematin, अब क्या होता है जब excessive heme production हो रहा है, we need only a specific amount of heme, right? अगर excessive produce हुआ तो the free heme जो होता है ना, free जो ज़रूरत से ज़्यादा, it will converted into hematin, and this hematin will allosterically inhibit synthesis of heme at the first step. So heme synthesis वहीं stop हो गया. Why? Because excess heme was being produced. This is again a type of negative feedback system only. The third and the most important one is compartmentalization. Now compartmentalization, how? See, the second, third and fourth step. This is the second step, this is the third step, and this is the fourth. This whole is the fourth step, let's say. This whole occurs in mitochondria. 2, 3, 4, just remember, 2, 3, 4 steps occurs in mitochondria. Okay, sorry, 2, 3, 4 occur in mitochondria. Sorry, cytoplasm, and the other steps occur in mitochondria. So basically, agar we stop the transport of something from mitochondria to cytoplasm, then the production ही stop हो जाएगा, obviously. Vice versa also. So अगर transport block कर दिया हमने, तो the cycle will be blocked. So this is another type of regulation of this cycle, compartmentalization. Okay, this is the regulation, all about the regulation. The last point in here is drugs and poisons. Now barbiturates, which are a type of drug, okay, what they do is they induce heme synthesis. There is a very beautiful logic behind this. So जब भी we intake drug, no, तो क्या होता है कि we have to detoxify these drugs, right? So body produces P450. I think you know where I'm going with this, right? P450 कब produce होगा? Obviously here liver will produce heme का ही तो derivative है ना P450. Now सारा का सारा heme is not converted into P450. 2/3 से is converted into P450. The other 1/3 is converted to heme, and therefore heme synthesis is induced, right? And the other point is that heme synthesis is prevented by high glucose levels in the body. So that is why when we learn about disorders of this system, barbiturates will precipitate the attack of porphyrias, right? Because porphyrias occur due to excessive heme synthesis. So barbiturates will increase them again, so it will precipitate the attack, so it will increase. So barbiturates should not be given in porphyrias, whereas a high glucose diet is given in porphyrias, which we will see the basis of. So high glucose diet will inhibit synthesis of heme. That is it for heme and its synthesis. I hope you like the video. Thank you.